Okay, folks, we're probably going to get going in the next minute or so. Just a couple of things. I want you to start thinking about your midterm. I know it's really early, but it's really not too early to start thinking about your midterm and doing well on your midterm. I am going to be around the entire midterm break, the the winter reading week. And if you are going to be around as well, you can let me know. We'll get a small group of students every single day if you want, or you can come every single day if you are really that hungry, and then we'll sort of do it informally. It's all about eating in this course, as you know, and we'll try to spend the time going through your lecture notes, making sure that you're on time, etc., and making sure that you're well prepared for the upcoming midterm. So we have like one or 2 minutes left. Do any of you have any questions or other things that you wanted to talk about beforehand? I'm going to take a little bit of Friday's class to tell you how the tutorials are going to be arranged and what you should expect in your tutorials that will start on Monday. But if you have any other questions related to the course material, I can also help with that right now if you do have any questions. Okay. Okay, so I'm going to go back and review and revisit a couple of things that we did earlier on in the course. And I'm going to go into a little bit more detail into some of the things that we looked at earlier on in this course as well. Some of this goes back to understanding genes as well as our behavior. So most of you, I would say the vast majority of you would say that being overweight, if you can possibly help it, and we went through some of the consequences, metabolic syndrome, and as you'll learn in your tutorial on Monday, that sets you up for neurodegenerative diseases. So things like Alzheimer's disease, and having meta-inflammation is a bad thing to have. And all of that is due to being overweight or being obese. It's not necessarily something that you choose to do. There are things that are now hidden in our food that we're not aware of. Sometimes it's just a matter of choice, and sometimes it's just a matter of convenience. So if you're really, really hungry and free food is being offered to you, are you going to turn down free food? As students, I would argue that you probably wouldn't. And that's a matter of convenience. Even if you know better if you're hungry, you're probably going to go ahead and eat food anyways. Whereas other things might not be our choice at all, and we'll go through some of that. We have a lot to cover today, and I'm not going to try to be disingenuous. This particular lecture, where we're going to talk about different aspects of genetics, you're going to see a fairly heavy representation on your midterm. So out of the 3 lectures that we've had so far, this one lecture here today is something that you should go back over this weekend and make sure that you're comfortable with the molecular mechanisms, some of the genetics, and other things. So I am going to go through some of the ideas that maybe we are predisposed. It's in our genes that we can theoretically gain weight. It's not only about choices that we make, it's also about the things that are directly encoded in our DNA. And part of the pathway involves leptin. And we know that from genetic studies that leptin is important in many aspects of weight regulation, food intake, and others. And I'm going to use one model that is not related at all to leptin. This is, again, I want to make this clear. Leptin is on a different chromosome. It's on chromosome 7. Prader-Willi syndrome is due to a number of things that happen on chromosome 15, but in many ways it kind of looks like there's a problem with leptin. And I want us to understand that sometimes weight gain is beyond our control, and it is directly encoded in our DNA. And then the last part I'll talk to you about is, you know, we talk a really good game about trying to manipulate the genome. We will continue talking about this in the upcoming lectures, but the reality is that many of those technologies that I'm going to introduce you to in this course are probably a few years away from being applicable in the context of regulating your eating. So along the way, I'm going to talk to you a little bit about mouse models. So mouse models are incredibly important to us because in many ways their genes are very similar to ours. We know that 300 or so different genes have been identified in mouse models that are relevant to different human diseases. And we want to be able to examine this in an animal model first before examining this in the human context. So we can examine it either on a behavioral level. Is the mouse eating more food? Has the mouse stopped eating more food? Or what is the molecular pathway in the brain that causes the mouse to do all of that? It would be very difficult to do those types of experiments in a non-intrusive way in humans. So we're going to take a look at some of these different mouse models and these model organisms in a little bit more detail today. Along the way, I'm going to go back, and one of the themes that I told you about earlier on was that in this course, we're going to learn the whole gamut of things from ethics all the way through to molecular biology, all the way through to behavior, and to some of the social implications of this. Now, this is physiology, and I know some of you took physiology, some of you have yet to take physiology, but I want to go back and revisit some of the basic tenets that are in physiology. Today, and throughout this course, one area of the brain that we're going to be looking at is the hypothalamus. When we are talking about the basic drives about maintaining your core body temperature on a cold day like today, your ability to eat, and a number of other core elements of your human physiology, we're talking about the homeostatic center. So for you to maintain your blood glucose, your core body temperature, all of this is regulated at the hypothalamus. And one of the things that's really important is many factors that are endocrine or hormonal can cross into the hypothalamus through the bloodstream and affect the hypothalamus and whether or not the hypothalamus is functioning normally. One of those factors is known as leptin. And we talked about leptin at the very tail end of the last lecture. And leptin is something that is normally released from adipocytes. It's released from fat tissue, and it's one of those factors that I mentioned to you earlier. And leptin is incredibly important in our discussions today. Leptin is a molecule released from fat cells. It is genetically encoded in your fat to release leptin. And when there is enough leptin in your bloodstream that will then travel up to the brain and signal to the brain, it acts as a brake. You stop feeding. So you don't continue to eat as much as you possibly can. Leptin is one of the mechanisms and one of the most powerful mechanisms that we have to suppress appetite. And again, if you're thinking about the different drugs that we'll be looking at later, being able to suppress appetite is one of those Rosetta Stone-like molecules that we want to be able to control. So we need to understand this molecule in a little bit more detail. How many of you have learned about adipocytes in some of your other courses? Can you tell me anything about what's going on here in terms of adipocytes? They're getting larger in size. Absolutely correct. So they store triglycerides, they store fatty acids, they take apart fat, and they store that in the center, in that sort of yellow region that you see here on this diagram. What else do you know about adipocytes from your other courses? So adipocytes are kind of unique. They do. Your colleague is 100% correct. They get bigger. But one of the things that you'll see is that adipocytes themselves are capable of division. And these adipocytes, when they get to a certain predetermined size, will start to divide. So they'll get to this size, for example, and instead of doing something else, they will decide to divide. Now, it's theoretically possible for you to actually make those adipocytes smaller. When you lose weight, those adipocytes that have been storing some of those fatty acids and triglycerides will get smaller. You will use some of those triglycerides and fatty acids, and we are going to see how you use them today. But the reality is, once you have an adipocyte, that stays with you unless you have so many adipocytes that some of them can die through apoptosis or necrosis, as we looked at in the last model on Friday with meta-inflammation. Now, this is where our story becomes kind of interesting. In the 1950s, in this huge warehouse down in the United States where they do nothing but breed mice for use in experimental studies, there was one mouse that was found, the offspring of this mouse, was found to have a spontaneous mutation. And this mouse just didn't stop eating. It kept eating and eating and eating, and it got bigger and bigger. And this mouse, because it became obese, was called this the OB mouse that you see down here. And this OB mouse, when they started to breed them together, so the mice got overweight after eating and continued to read was found to have a mutation in what was then known as the OB gene. Later, it was found to be on chromosome 7. They didn't know this at the time in the 1960s. Much, much later in the 1980s and into the parts of the early 1990s, it was found that this lack of a gene, or this mutation in a gene, actually resulted from the fact that this mouse kept getting bigger and bigger and fatter, but the reason why it kept eating was that it didn't produce leptin. So the adipocytes kept getting bigger, and adipocytes are normally supposed to release leptin, but in this case, the adipocytes did not have the OB gene, and it could not produce leptin, and as a result, there was no leptin signaling. That was the first clue that perhaps leptin is the control that suppresses appetite, because this animal certainly did not stop eating, and this is sort of an age-matched control. You can see it's much larger in size as well as being overweight. Later on in the 1980s and 1990s, as people started to dissect the molecular pathways that were involved, it was found that there was another mutant mouse that did not have this issue with leptin in its bloodstream. It had normal levels of leptin, but there was a problem in the receptor for leptin, and this was known as a DB mouse. Either one, either having not enough leptin or not having the leptin receptor, in other words, a disruption in the leptin signaling pathway into the hypothalamus led to the mouse being overweight. So this is where our story comes into play. So going back to classic physiology, and some of you will have remembered this from physiology, and I'll go back over exactly what you need to know. You don't need any extra material from physiology. I'll give you sort of our version of physiology. As your adipocytes start to pack away the extra fatty acids and triglycerol, and as the cytoplasm gets bigger and bigger, after a meal, for example, or as you're gaining weight, more and more leptin, in theory, should start to be produced by these adipocytes and get into the bloodstream. That leptin then travels in your bloodstream and will then target the hypothalamus and certain groups of neurons in the hypothalamus that will then turn off your feeding behavior. There are neurons that are there in your hypothalamus that basically want you to feel hungry or want you to start eating. And we'll see this a little bit later on, and you can write this down and go back and reference this later. On slide 19, we'll go into this in a little bit more detail. Eventually, when leptin gets to the hypothalamus, it turns on a group of neurons, which then releases a substance that on this slide is called melanocortin, and melanocortin suppresses your appetite. And we'll see that later, this substance is also known as POMC, or a longer form of this melanocortin. Do you have any questions about this before we go on? Okay, your hypothalamus is being targeted, leptin is there, and this will suppress your appetite. So what happens if the reverse is true? Why do you actually eat in the first place? Again, there are many, many reasons why you will eat. Some of you will know that from physiology. This is one mechanism out of many, but this is a very powerful mechanism. Again, just out of curiosity, it's about 2 o'clock or 2:15-ish. How many of you are usually eating at this time of day? A couple of you? How many of you are hungry right now? Okay, even though you're not normally supposed to eat. So if you're really hungry, I could give you a healthy choice or a non-healthy choice. How many of you will opt for a healthy choice? No one. Okay, that's great. And so I failed you as a prof. That is a terrible thing. I have very unhealthy choices for you, and I'll kind of leave it up to you what you decide to go ahead and eat. But some of it, as I mentioned to you, this is physiology. This is molecular biology. Some of it is really not your choice. If you're really hungry and you know it's unhealthy, but you just want to tide yourself over, you'll eat it, right? Even if you know better, and it's kind of better than going hungry. And so my favorite chocolate that you'll see me eating all the time is Twix. Yes, it's really good. Or you could have a nice tangerine or whatever this is. Or you could decide to have a quinoa granola bar of some sort. And I'll just pass this around. Someone want to come and grab it and then maybe pass it around? Yes? Okay, thank you. And you can take whatever you wish. No judgments. Okay. But what happens when you are actually hungry? And I want you to remember this pathway as well. when your adipocytes are actually not storing the not storing triglycerides or not storing free fatty acids, and you are starting to get the adipocytes shrinking in size because they increase and decrease in size, then the amount of leptin that is being produced by these adipocytes actually start to drop, especially if you are fasting. If you are fasting or you're waking up in the morning and you're feeling really hungry, your leptin levels have decreased quite substantially. And when leptin levels in your blood start to decrease, as denoted by the smaller number of these little leptin molecules, then without leptin being there, the hypothalamus actually is now capable of releasing another substance which is known as neuropeptide Y. And in fact, later on slide 18, you're going to learn that this is just one part of the story. There are other aspects of this as well, not just neuropeptide Y. There are other factors that also get released that stimulate hunger. So you might want to refer later to slide number 18. So for us as human biology students, we want to be able to manipulate the system. And it happens physiologically when you're hungry or when you're feeling full, leptin levels rise, leptin levels fall, and you eat or you decide not to eat as a result of leptin and or neuropeptide Y levels on a very simplistic level. Again, there will be more levels of sophistication in a moment. So your adipose tissue, in other words, and this pathway was worked out in the early part of the 1990s, when your adipocytes are at a certain size and they are releasing leptin, this leptin will then go via the bloodstream, and because of its relationship to the hypothalamus, it is capable of targeting different groups of neurons within the hypothalamus. The hypothalamus is just like one massive structure. It's like saying the brain, right? So you know that there are different regions in the brain. There are different groups of neurons in the hypothalamus. One of the ones that are going to be the most important for you to know today are the groups of neurons that are found here in the hypothalamus, known as the arcuate nucleus. So the arcuate nucleus will contain a certain group of neurons that we'll talk about in more detail later. And we'll also talk about the paraventricular nucleus, and we'll, for the time being, ignore the ventromedial nucleus. And these are where the leptin receptors reside. The leptin receptors are here on the arcuate nucleus, okay? Neurons that are found in the arcuate nucleus. So we release more leptin, we release less leptin, we change the levels of neuropeptide Y, we decide to eat or not eat, all related to the firing and the release of different substances like melanocortin here at the level of the arcuate nucleus, which will then target other groups of neurons. Do you guys have any questions before I go on? So why is this all important? So this is important for a variety of reasons. One of the reasons is if we are releasing leptin, which means that we have eaten enough, we don't need to store more energy under normal circumstances. One of the things that happens is that through the hypothalamus and through what is known as the nucleus tractus solitarius, or the NTS, and one of you were asking, will you get, if you mention a short form here, will you also mention it on the midterm exam? I will give you both the long-term name, the nucleus tractus solitarius, and the NTS on your midterm and final exam. So in case I mention it one way or the other, you'll know what I'm talking about. Through this sympathetic pathway, one of the things that happens is it signals back to those adipocytes. And these adipocytes are really important, again, in this regulation. So it's a negative feedback loop. I've eaten enough. It's now left, leptin has left the adipocyte. It's signaled to a group of neurons here to stop eating more. And it's also signaled to the sympathetic nervous system, to the nucleus tractus solitarius, back to the adipocytes. And the sympathetic nervous system releases norepinephrine or noradrenaline. And one of the things that happens is on these adipocytes, that is now exposed to the norepinephrine being released from the nucleus tractus solitarius, this beta adrenergic receptor, the beta 3 adrenergic receptor on the adipocytes responds. So leptin has left, and now this other signal has come back to the adipocyte. Why is that important? This is important for two reasons. One of the reasons why we eat, if you're not aware of it, is to actually maintain our core body temperature. Without this ability to maintain our core body temperature, you will suffer from hypothermia, not severe hypothermia, but your body temperature will start to drop. And one of the reasons why this happens is this beta-3 adrenergic receptor binds to norepinephrine that is being released. And one of the things that happens is that it initiates a intracellular signaling pathway through the G protein coupled receptor that does a couple of things. One is that it will activate cyclic AMP or form more cyclic AMP by turning on adenylate cyclase, which then turns on protein kinase A. And this turning on of protein kinase A is really important because, one, it is going to signal to the nucleus to start increasing the production of uncoupling protein. And this uncoupling protein is important in the mitochondria for beta oxidation. And the mitochondria is what allows you to actually produce heat. And how does it produce heat in beta oxidation? It burns the fat that has been stored in this adipocyte. And so this becomes a really important pathway. And how do we get more fat that has been stored in this adipocyte available for this beta oxidation? This same protein kinase A in this adipocyte that is now being turned on by norepinephrine will also activate enzymes like perilipin that will then chew up and release any of the triacylglycerides that have been stored back into free fatty acids that can undergo this beta oxidation that you see here. And this uncoupling reaction produces the heat that allows our bodies to stay at 37 degrees. So if all of this were true, and again, you'll have to take my word for it that in mouse models, much of this has been worked out, do we actually see individuals that have problems with their weight where there is a deficiency in this leptin signaling pathway have all of the issues that we would see here? And in fact, in a very small number of individuals with childhood forms of obesity, a very small number that have what is known as leptin deficiency, in 2 to 10% of individuals with childhood obesity, leptin deficiency leads to weight gain because there is no leptin to signal back, and they are very, very cold all the time. They have this inability to stay warm. And we know that these pathways are linked together through this molecular pathway. And again, I don't believe in being disingenuous at all with you. Your time is really valuable. Plus, I want you to be able to learn things. This is a pathway that you should expect to see in some shape or form on your midterm. And I am actually going to show you on Friday, I'm going to take 5 minutes, how I would write a multiple choice test question on this. It is not the type that I'm thinking that most of you will expect, but by the end of Friday, you should know like what style of question I might ask you based on this. Now, the other thing that I haven't shown you here is that I told you that the OB mouse has problems with leptin release. The DB mouse has a problem with the leptin receptor. Now, we won't have time to talk about the leptin receptor, but I can tell you that in some individuals, there is some evidence, just like insulin resistance, that there is leptin resistance. If there's too much leptin around all the time, perhaps in some forms of obesity, that the cell just simply stops responding. It doesn't undergo this beta oxidation at all. Now, I'm going to take a little bit of a side tour here, and I'm going to talk to you a little bit about a very different type of genetic disorder that leads to identical behavior, overeating. And this is what is known as Prader-Willi syndrome. Have any of you heard of Prader-Willi syndrome, PWS, in some of your genetics classes, hopefully? So I'm going to tell you that, again, right off the top. I told you that this is a lecture that you should spend some time on. You will also be tested on the basics that you see here on this slide related to Prader-Willi syndrome. So in Prader-Willi syndrome, unlike leptin, which is found on chromosome 7, and there's no slide for that, but hopefully you've written down somewhere that leptin is found on chromosome 7 or the OB gene is found on chromosome 7, here we have issues with chromosome 15. And in particular, chromosome 15, the Q arm or the long arm of chromosome 15 at locus 13. And whether it's turned off by epigenetic mechanisms or whether you get 2 copies of the maternal the maternal gene and there's imprinting that's going on there, something happens where the correct expression of genes that are found on this chromosome are turned off. Now, why is Prader-Willi so important for us in our discussion? In Prader-Willi syndrome, these individuals just can't stop eating. In fact, they will eat themselves to death. This is a very severe behavioral problem. If you are someone who has a child with Prader-Willi syndrome, you can't actually leave your refrigerator unlocked. They will lock up the refrigerator, and guess what? The child that you have, because they're so hungry all the time, will actually find ways to either break into your refrigerator, will steal money so that they can buy food if they can't break into your refrigerator, or engage in all kinds of behavior to get the money to be able to buy food. And this excessive eating will lead to not just obesity, but we're talking life-threatening morbid obesity, simply because something on this gene that is not related to leptin causes them to eat and overeat. Now, it doesn't start off that way. It starts off in early infancy where they look very normal and they have problems actually eating. And feeding difficulties in early infancy. Their muscles tend to not be well developed. They have hypotonia of their muscles, no muscle tension or lower muscle tension. And we know that there are lots of issues associated with it. They will die from morbid obesity unless this is controlled. They have heart disease, heart issues in their teenage years that make them look a lot like they're in their middle-aged years. So we don't understand all of the genetic pathways. But we know that eating and excessive eating is something that is a behavior that sometimes is beyond our ability to control. Now this goes back into leptin. So this is more like the OB mouse without leptin signaling that I showed you earlier. What happens here is that these individuals are born with a leptin deficiency. It is a genetic condition in very small numbers of individuals. About 2 to 10% of overweight children have this. And their adipocytes don't have the correct gene to be able to produce leptin, and they simply keep eating. There is no leptin signal to say stop eating. And so as a result, they keep eating all the time. They have issues with morbid obesity as well. They also have other issues. And we know from this, and this is something, again, that you should be aware of for your midterm, that leptin does not only affect the systems that I mentioned to you. The leptin signaling cascade will also affect the immune system. It also affects bone density. It affects sexual maturity, etc. Leptin receptors are found in a variety of different tissues, even though I'm concentrating on the hypothalamic system. So again, they have infertility issues where they aren't forming proper follicles. They have lack of gonadotropin releasing hormone. All of these factors come as a result of not having enough leptin in their blood from those adipocytes. Even though they're accumulating more and more adipocytes, there is no leptin for them to turn things off. Let me ask you, what do you think was done to this individual to cause this same individual to go back to a normal metabolic status? What do you think was done to this individual? So their adipocytes have a problem with producing leptin, and without that leptin to turn off the feeding behavior, they can't suppress feeding, they keep eating. So what do you think was given to this individual? Yes. Sorry, leptin. Injections of leptin, that's right. And so by just providing that leptin, it is enough for them to actually stop eating. And so there's no deficiency in the receptor. It's only the fact that they don't make enough leptin in the first place. So this is, again, a really important slide for you to know. And in some ways, it's not this slide. It's the slide that follows after. But this is a larger view of what leptin and leptin signaling is. Remember, leptin is being released from the adipocytes. It is traveling all the way up to the hypothalamus. So what you're actually looking at is the plasma membrane of a neuron in the hypothalamus that is now responding to leptin. And within certain groups of neurons in the hypothalamus, when leptin binds to its receptor and its co-receptor, and they're again, are differing opinions on what this co-receptor is. One of the things that happens is that these will dimerize. They will come together, as you see here on the right, in response to having leptin present. And this particular enzyme that is normally found associated under resting conditions with the leptin receptor now becomes activated. And this enzyme that is really important is known as the Janus kinase, or the Janus kinase 2 enzyme that you see here. It's also called JAK2. Okay, so Janus kinase 2 or JAK2. And now when these things dimerize, they become activated. And when the kinase is now activated following dimerization, the leptin receptor itself is phosphorylated. And again, the details are not important. Which residue is not important at all. But these tyrosine residues become phosphorylated, which then acts as a signal for other molecules to come up. And one of those molecules that come up to this activated leptin receptor is known as STAT3. And this STAT3 molecule is normally found in the intracellular region. And the STAT3 molecule contains what is known as a Sark homology domain, which causes it to bind up here. And one of the things that happens is when this complex is activated by the presence of leptin, one of the things that happens is that each of these STAT3 molecules in turn becomes phosphorylated by this Janus kinase. Okay, so it's this JAK2 that causes the phosphorylation of the leptin receptor as well as the phosphorylation of STAT3. Is that clear for everyone? Yes? No? Kind of? Need time? I need a little space? Need a little time? Figure it out later on my own? Okay. So why is that important? This is important for a couple of reasons. On STAT3, once it is phosphorylated, it has this unique capacity to form a dimer on its own. Usually these dimers form and then they got phosphorylated. These dimers don't form in the case of STAT3 until they are phosphorylated. When we get two STAT3 molecules that are phosphorylated, they form this dimer, which then translocates into the nucleus, and it will turn on the transcription of certain types of genes. Now again, I don't need you to go nuts memorizing all sorts. In fact, there are a multitude of different genes, but the ones that are going to be important for us in this course, and the ones that I need you to memorize, and you don't have it written down, and in case you can't write them down right now, this will be available on the YouTube stream. One of these is the pathway known as SOX3, and today we'll see this POMC, or the pro-opio melanocortin gene that turns on the melanocortin neurotransmitter that I mentioned to you earlier. So all of this is occurring within the hypothalamus. Is that okay for everyone? You should know this pathway. And again, we'll work through some of these in the upcoming lecture on Friday on how I'm going to be testing you on it. And this weekend, even though I know you're going to be busy, I'm going to start giving you some practice questions in the style that I will be asking you on the midterm to try to get you ready. And you can get ready, you can do really well on the midterm if you give yourself enough time to study the details like these details that I'm sharing with you here. This other molecule is kind of a neat molecule as well. Once we started to realize that leptin can actually be involved in a number of different signaling pathways that lead to your weight control and being overweight or not, other molecules, especially ones that were released from adipocytes, were also screened. And one of the first molecules to come out of this adipocyte screen is known as adiponectin. And adiponectin is a huge protein, not huge in terms of size. We have a lot of it in our bloodstream. In theory, in a nice healthy individual, we have lots of this adiponectin floating around our bloodstream all the time. And adiponectin is kind of unique. It does a number of things. It helps to sensitize tissues that require insulin. So in some ways, it almost acts like insulin, driving insulin activity. And it has some anti-inflammatory properties. So you can see some of the benefits of having lots of adiponectin around. Some individuals, and you can go back and read the Trujillo paper back in 2005, which was a review of all of these effects, also lead to anti-atherosclerotic properties. So it reverses atherosclerosis, or the buildup of plaque in different blood vessels. So adiponectin is also released by the adipocytes, but it actually has an inverse relationship. Leptin, as the adipocytes get bigger and bigger, can theoretically release more and more leptin into the bloodstream. But here, the more of these adipocytes you have, the less adiponectin you will actually release into the bloodstream. So it has this interesting inverse relationship. And some of the reasons why people are interested in this is because when you start increasing adipose tissue in metabolic syndrome and you start decreasing adiponectin, it might start to interfere with your cardio metabolic effects. So your heart will start to be affected. And we know that metabolic syndrome isn't just you gaining weight. It's you losing insulin sensitivity. It's problems with your heart and other factors. And so one of the things that we are interested in with this adiponectin is, how does it actually work? And can we create synthetic compounds that might go back and start to reverse some of the damaging effects of having an ischemic heart? So it might prevent this ischemia from a damaged heart that accumulates when you have metabolic syndrome, maybe through the inhibition of inflammatory processes. It might prevent cell death by activating AMP kinase pathways, increasing energy availability, and preventing or blocking cell death in the heart from occurring. Or it might lead to the activation of an enzyme known as cyclooxygenase, or COX-2, which will then produce more prostaglandin E2, which might turn off some of those inflammatory responses that we talked about earlier, things like turning off TNF-alpha production, which will also damage your heart. And so maybe this is the way in which it works. And individuals in pharmaceutical companies are interested in trying to turn on this pathway in these different types of adipocytes. So I'm going to go back and tell you a little bit more about the hypothalamus. Your drive to eat is somewhat hormonal, so leptin is one, but there are other signals, as I mentioned to you earlier. Some of it is purely behavioral. Some of it is, if you're eating and you happen to be with friends, you'll eat more. Some of it is really time of day, other types of behaviors similar to that. Some of it is driven by your senses. And we'll go through some of those different mechanisms, including the control or lack of control if you have this pathway, which is the hedonistic pathway. Like I really feel like I want to eat something sweet or salty or fatty a little bit later on in this course. So the brain is actually acting as an integration center that allows you to maintain this homeostasis that I mentioned to you earlier, particularly related to blood glucose. Your hypothalamus sits right here, and it is in communication with other brain centers that will leave the brain, like the nucleus tractus solitarius or the NTS, that sympathetic pathway that I showed you earlier. If we took the hypothalamus, which is exposed to the blood, and so you can imagine blood vessels running by, leptin coming in and targeting this area of the hypothalamus. If we took a look at the hypothalamus, we would see an area known as the arcuate nucleus that I showed you earlier. And within the arcuate nucleus, one group of neurons, in this case color-coded, sort of this orangish red color over here, this pathway, if you stimulate this pathway, it will turn on your feeding behaviors. And when these groups of neurons that control feeding behaviors are turned on, they do so by releasing appetite-stimulating neurotransmitters like AGRP, or the agouti-related peptide, as well as neuropeptide Y. And we talked about when leptin falls and when there is no leptin signaling occurring in the arcuate nucleus, these neurotransmitters are now being released more and more frequently, and as a result, they will stimulate feeding behaviors. The other group of neurons that you see down here are also really important. And these are also found in the arcuate nucleus. And this group of neurons releases a different set of neurotransmitters. And these are the N-orexigenic. Orexin is a molecule that causes you to eat. Anorexia means that you don't eat enough. And this is a non-eating pathway, an anorexigenic pathway. So this group of neurons in blue, if you stimulate them, they will actually cause the release of neurotransmitters like the cocaine and amphetamine regulated transcript, as well as the melanocortin that we talked about earlier, or the pro-opio melanocortin, the full name for it, that will then act as an inhibition for feeding. And so when leptin is signaling here and turning on the transcription of POMC, it will cause POMC to be released. It will activate other hypothalamic neurons, and you stop eating. And again, it's useful to go back to put this in context of slide 9. All of this, in theory, is how you eat and stop eating. But the reality is we are controlled by a number of other factors beyond this. So at this point, to be honest, I like eating. I like eating a lot. I like eating. I don't eat 3 times a day, which is actually what I should be doing. But when I get home, instead of eating dinner, I have a pre-dinner snack. I have dinner, and then I have an after-dinner snack. Yes, as I said, this class is all about gluttony. And in fact, I know better, and I know that I should be taking better care of myself, but the reality is that I am not at that point where easily, through normal sort of behavioral modifications, that I can control my weight easily. And so I think about this a lot. So should I actually start taking drugs that might control my weight? And there are a number of different drugs, and I'm going to go through what you need to know. You do not, by the way, have to know any of the structures, nor think that you have to draw out these different structures, nor do you have to know the manufacturers for any of these drugs, nor when they're launched, or how many millions of dollars they have in sales. But I want you to get a better sense as to how we try to control weight. You're not able to control yourself behaviorally, neither am I, and it becomes really bad. So some of you will have had that Twix bar knowing that it's not something that ultimately, I can tell who ate it because you're looking down in shame or looking off to your friends. It's okay, I love Twix. If you're really hungry for Twix, drop by my office anytime. We'll have a Twix feast. So if you're not able to control your diet and you're not able to control your behavioral intake, maybe you should start taking some of these if you're really physiologically in danger of being morbidly overweight or obese, different types of drugs. The first class of drugs is this one that I'd like you to make sure that you know. We're going to learn it by the name orlistat. And orlistat, what it does is it blocks the absorption of fat from the gut directly. So even if you had a fatty meal, your fat, that fat that gets into your bloodstream is going to be blocked because orlistat blocks a lipase that prevents you from absorbing any fat. And that's one way drug companies work to control your weight. But look at all of the other classes that I'm going to show you in the next few minutes. So this drug here, sibutramine, is a really powerful drug. So is the one below it. And again, we won't worry about all of the details. All of these work on your brain. They're working on your neurotransmitters by changing the levels of catecholamines, and as a result, changing how you feel in terms of feeling full or whether or not you should be eating. All of these, by the way, have side effects associated with them. Side effects, again, that you don't need to know in a lot of detail, but these are severe side effects. So this other drug here, rimonabant, is actually a little bit different, but many of you will recognize CB1. What is CB1 in this context of looking at the brain? Do any of you know this from any of your other classes? Because we are still affecting the brain in this class of drugs. CB1 antagonist? Have any of you heard of cannabinoids and cannabinoid receptors? So the cannabinoid system, which is being targeted here. So some of you will have heard that if you take marijuana, for example, or you smoke marijuana, that you have this phenomenon known as having the munchies. Like you feel like you're eating after you smoke. And a great movie for you to watch if you're really interested in that whole phenomenon is Harold and Kumar Go to White Castle. That's a great movie. It demonstrates all of the munchiness associated with pot use in graphic detail. And one of the reasons why is that this cannabinoid receptor, if you turn it off, it will actually suppress feeding behavior as well. But these all, again, have severe side effects. Rimonabant was actually taken off the market briefly because individuals that took it, 900 cases worldwide, those individuals who wanted to suppress their diet and lose weight that took Rimonabant actually wanted to commit suicide. And it was real suicide ideation. They went out and they started to really fixate on this, and it was taken off the market briefly as a result. But look at all of these other classes of drugs. If you were asked to design a drug that would help you lose weight, in fact, one of the things that you would do is really look at drugs that affect your brain, whether it is a neurotransmitter level, something that looks like an antagonist of a cannabinoid receptor, something that looks like amphetamine. All of these are targeting your brain. And again, these last few that you see here, outside of Rimonabant, you don't have to know the names. Just be aware that the vast majority of drugs that are either in use or being developed affect different neurotransmitter systems in your brain to affect behavior. So I'm going to briefly talk about some of the background into the next paper. All of those drugs that I mentioned to you earlier affect the brain. All of those drugs will, in one way, even with the side effects, change the way in which you ingest food or if you feel hungry, etc. All of those drugs sell extremely well worldwide because we care about our weight. We know it's an epidemic in terms of being overweight or being obese. But all of those drugs that I showed you, whether it's Rimonabant and suicide ideation or having nausea or headaches or other types of side effects, all of them, every last one, even the ones that have been on the market for over 30 years, have severe consequences and side effects. People are always looking to look at this pathway that we've just looked at and to see if there are other pathways that we might be able to manipulate using other drugs that might potentially have fewer side effects. In your brain, in order for your brain to be nice and healthy, we have these other proteins which we call neurotrophins. It's like having fertilizer around for your brain cells. Your nerve cells grow, they have dendrites, and they will spread out more dendrites. They'll be healthier if you have these neurotrophins. These neurotrophic molecules, or neurotrophins, also act just like the leptin signaling pathway through tyrosine kinases. They will be phosphorylated on these receptors, and as a result, because they look very similar to receptors that are already in the hypothalamus, people wanted to know, is this pathway also involved? Because it's correlational, it kind of looks the same, and it's found on a number of different areas like the hypothalamus. It's been shown in other studies that maybe it suppresses appetite if we use one neurotrophin, which is called BDNF, or brain-derived neurotrophic factor, and it signals through its TrkB receptor, TrkB, that when you stimulate this in mice, our mouse model that we looked at earlier, mice are incredibly important, that when you stimulate areas in the hypothalamus, it also suppresses appetite, it reduces body weight. If you go back and look at the genetics, there is a knockout animal of BDNF. No more BDNF, just like no more leptin. If we take a heterozygous animal, so the amount of BDNF is decreased. It has one copy, but not the other. In these animals, these tend to overeat. So not having BDNF causes overeating, or not having enough BDNF causes overeating, and activating BDNF causes you to eat less. It sounds a lot like leptin. And BDNF is something that's in your brain. How many of you have heard of BDNF? A couple of you? If I wanted to stimulate BDNF in your brain right now, what would I do? Any ideas? Because you hear about it in the news all the time. How do I increase BDNF levels and therefore maybe suppress appetite by increasing BDNF in your brain? Any ideas at all? Have you heard of exercise? Yes? Exercise, in theory, increases BDNF in your brain, and exercise has been shown to control weight etc. So we'll talk a little bit more about that a little bit later on. So right now, people are looking at whether or not neurotrophins, exercise and BDNF, for example, is that one way in which it controls your weight through these TrkB receptors. We know that these neurotrophins, like BDNF and their receptors, are expressed in a whole bunch of metabolically active tissues, not only in the hypothalamus and not only in the brain. And if we knock out one of these receptors, the P75 receptor, the P75 neurotrophin receptor, it actually protects mice from developing obesity and metabolic syndrome. And this is what we're going to be talking about in the next lecture. So how can we, using mouse models, understand more about this process and also understanding the bigger sort of human biology aspect of it? Nobody wants to be injected with chemicals. Nobody wants to be injected with viruses. Nobody wants to have issues where where there's gene delivery or genome editing. But if this pathway is true, and the other pathways are true, is it possible for you to really lose weight, not in the conventional sense of losing weight by burning muscle, burning fat and developing muscle, but also directly through the activities of TrkB? And if that's the case, exercise really is the best sort of medicine as well as drug. Now, I want you to go back and just highlight things today. Don't bother memorizing them now. Hopefully over the weekend you'll have some time to do this. Go back over the leptin pathway, look at Prader-Willi syndrome, leptin deficiency, and then the drugs, where they work and why, and go over the arcuate nucleus and the different neurotransmitters that are associated with each. And I'll talk to you more about this pathway on Friday. Hello.